Brian, I have your quiz. And the lab notebook, I have it. He didn't come to lab last week, so we didn't pick it up. He comes last week.
Yeah.
And so this is a little.
No, they did it on.
That is not stated. And then there's your. You are. I'm gonna.
Okay, yeah, I checked on it, like, I spent so much my other classes.
Yeah.
We can still do like that. I just
Okay, so let's get started today. So remember that your midterm is Wednesday.
Did you turn the recorders on?
I did.
Thank you.
During regular class time, did you hear? Okay, so you have this, okay, we have these. So you need to. You can do this, which now is, and, are you, is there a reason why you're gonna have that? Okay. So you do on Thursday?
Yeah.
Okay, Thursday. Okay, so you have your midterm on Wednesday, and we have the lab today, which is going to cover cell reproduction. So the last thing that we're going to be doing today is our last thing before the midterm is we're going to be covering chapter 7. So the information in chapter 7 deals with cell reproduction. So we already talked about the cell cycle a little bit, and so do you remember in the cell cycle, what does M stand for? Mitosis. What does the G stand for? Growth, right? So the cell has to produce maybe more organelles, more proteins. And then S stands for synthesis. So what is being synthesized? DNA, right? So the DNA gets copied, it gets replicated in the S stage of the cell cycle. So remember that cells can leave the cell cycle and become actively not dividing, so not active. So they can become specialized. So for example, fat cells can become specialized and just get larger or smaller depending on how much triglycerides are in them. And we can also have bone cells, and we can have nerves or neurons, right? So those are specialized cells that generally do not undergo reproduction very often. Okay. So, today we're going to be looking at chromosomes, and so I need to talk a little bit about karyotypes. So when do you get a karyotype? Has anybody ever had the opportunity to have to get a karyotype analysis? Anybody know? So one of the reasons why people have prenatal testing, and amniocentesis in particular, is that during amniocentesis, they take out some of the amniotic fluid, and there are fetal cells in the fluid. And they grow those fetal cells outside in a petri dish, and then they do a karyotype analysis on them. And so the karyotype is specifically looking at the number of chromosomes and the shape and size of the chromosomes. So number.
Is that the test that you did for Down syndrome?
Yes, shape, size, and then they can also look at what is called the banding pattern. And this is all of chromosomes. Okay. So our cells that are our body cells are called somatic cells. So these are just normal body cells. So like our fat cells, like our neurons, like our muscle cells. Those are all somatic cells. And they have, in humans, we have 23 pairs of chromosomes. That's a 3. 23 pairs of chromosomes, or a total of 46. So the pairs of chromosomes have to do with chromosomes that are the same size and the same shape, right? So if we look at a chromosome and it looks something like this, remember we talked about the centromere is the middle of the chromosome, and then we stain it with stain and the DNA picks up the stain, right? So it has a particular banding pattern. If chromosomes are the same size and the same shape, they are said to be homologous pairs. So if I drew another chromosome that was the same size and the same shape and had the same banding pattern, right, I would say that these two chromosomes are homologous. So homo means same versus hetero, which means other, right? So homologous chromosomes mean that they have the same size and the same shape. They have the same banding pattern, and they also have the same genes. So I mentioned before that genes are just pieces of DNA on a chromosome. So these would have the same genes. You know, and I drew them a little bit differently, but when you actually look at the picture, sometimes the chromosomes are like, they have little kinks in them, and they might be a little bit different shaped. But a person who studies karyotypes can sort out chromosomes like this. If I gave you just your 46 chromosomes and you had to match them up based upon the size and the shape and banding pattern, it would take you a long time to do so. But a genetics counselor can do it really, really quickly, okay? So if we look at a karyotype, so let's say, for example, generally if you are over the age of 30, I can't remember, 35, I think, oftentimes they'll offer you an amniocentesis to make sure that the fetus has the proper number of chromosomes. And one of the things that they're particularly worried about in older females is Down syndrome. So if you look at this is not obviously the way that it comes out when they're actually staining them, right? They're all kind of like, big bunch. And then the genetics counselor sorts them out and then pairs them together. So chromosome number 1 is always the largest chromosome. And then you notice that this pair down here, 22, is the smallest chromosome. So they're numbered based upon their size. So these are homologous pairs. So if you added all of these up, there would be 46 of them. These chromosomes are what we call the sex chromosome. So would this individual be a male or a female? Male. And the reason why they're male is that they have one X and one Y. And the X and the Y, we'll talk about this later, you don't need to know this right now, but the X and the Y are not homologous. They're not the same size and the same shape, and they have different genes on them. In fact, there's a little region on the Y chromosome that will determine whether a baby will become a male or become a female, right? So if it's present and it's normally functioning, then the fetus will become male. Okay? So this is what is called a karyotype. Now, if you look at a person that has an extra copy of the 21st chromosome, and so if there's another 21st chromosome, that is what would be Down syndrome. So when we say that,
So a third copy.
a third copy. So if a normal person, this karyotype right here would be 46, and it would be XY. Right, so I would say that there's 46 chromosomes, and this is a male, okay? If there's an extra chromosome, like with Down syndrome, I would go 47, and I would go plus 21, and then I would go XY, right? So what this tells me is that there's one extra chromosome, and it's the 21st chromosome. So it could be 47 plus 13, which sometimes happens, or 47 plus 18, I think is the other one that sometimes happens. But this is a really common one. And so this just tells me it's a male with Down syndrome, right? So it's a karyotype analysis. Now they've gotten really fancy with karyotype analyses now, but like 11 years ago, I had one done for my baby, and the geneticist actually sent me a picture of the karyotype, and it looked just like this, right? But when now they have a little bit fancier techniques where they can actually stain the chromosomes with fluorescing dye. And so this makes it much easier to sort them, obviously, right? So if this is all the chromosomes, I can just now match them up. And it has to do with the dye binds to specific regions on those chromosomes that are found on those chromosomes and nowhere else. Okay, so it's a little bit fancier. So this would be a female, because you notice that the two chromosomes are about the same size and the same shape. This one's squashed a little bit differently, but it wouldn't be a tiny one, right? So this is a female. And then you can actually do some more chromosomal analysis.
Is that for both X's, or is it just?
Those are two X's. Yeah, so two X's. There's no Y. And then on this one, what they're doing is they're looking for a particular sequence of DNA. And so the yellow shows me where the sequence is. And so we now kind of fluoresce the chromosomes to see if there's any abnormalities on them. So rather than just staining them with dye, we can we'll talk about this more when we get to genetics and the inheritance of chromosomes. Okay, so that's karyotype analysis. So when we look at mitosis, right? Actually, I wanted to go back here, sorry. I want to go back to this idea of karyotype analysis. Okay, so those are somatic cells. So remember that we talked about gametes. So gametes are egg and sperm. Right? So these are special cells that only have a total of 23 chromosomes. So there's a total of 23 chromosomes. And so in this case, there are no pairs. Okay. So what this suggests is when the egg and the sperm come together, one copy of the homologous chromosome comes from your mother, and one copy of the homologous chromosome comes from your father, right? So when you look at these chromosomes in your textbook, you're going to see that they are colored. So they use red and blue, right? So they would like the red, so I'll just put this in parentheses, the red chromosomes are maternal in origin. And then the blue chromosomes are fraternal, so they are father.
So the
Oh,
color,
paternal, sorry, paternal.
the colors, is that in like most documents? Is that how they do it?
Yes.
Most don't have colors, so.
Oh, yes. Oh, that's a problem.
Yeah.
But I'll show you some pictures.
Okay.
And we're actually going to use red and yellow. In
Well,
the-
on the online one it does.
So this is paternal. Yeah, online they've been colored. Okay. So the chromosomes that are found in the sperm, right, are going to be blue, and the chromosomes that are found in the egg are going to be red, and then they come together, and so you have chromosomes from each parent represented in your somatic cells. Okay. Now, because these gametes are only 23 chromosomes and no pairs, they are said to be haploid, or 1N, where N equals the number of chromosomes. So you'll see that term, haploid, and then you'll see 1N, right? So that just means that they have one copy of each chromosome, so there are no pairs, okay? So if we go back up here to the somatic cell, because we have two copies, this is said to be diploid, and it is 2N. So our body cells are diploid, and they are, pen's not letting me do that, 2N. Okay. Okay, so now we can talk about mitosis. Yeah. Oh, my word, this is not staying in place today. So there's 23 pairs of chromosomes, a total of 46. There's a red one and a blue one. There's 2N, and they are diploid, which means di means two copies, versus haploid, which just remember hap means half, so that they have half the genetic material in gametes as compared to the somatic cells. Okay. Okay, so when we go to mitosis, this is where we go from a diploid cell, one diploid cell, right? And we produce two diploid cells. So one diploid cell, and then we get one diploid.
So I have a question then.
Yeah.
So then, on the karyotype things that you were showing, and have the pairs of homologous chromosomes.
Yep.
So then, with the gametes, and they're only having, one of each of those, plus either an X or Y. And then when it joins with the sperm, and they have, so then are those other, besides the Y, or the other, anyway, are the other 22 of them, will they be identical, like would they be homologous?
They would have the same genes, but not necessarily the same form of the gene. So we'll talk about that when we get to genetics. They're homologous because they have the same genes. So you get half your genes from your mom and half from your father, and they are the same genes, but they're different forms of the gene that gives you the differences that you see in the offspring. And those different forms and genes are called alleles. But we're going to talk about that after the midterm.
But the one would pair up with the one, and the two would pair up with the two. They would be the same gene, but maybe a little different, right? So your mom could have red hair gene, and your dad could have the brown hair gene, right? But it's the same gene. Okay, so this is where we go from one diploid cell, right? And then we divide and we produce two diploid cells, because mitosis is a type of cell division that is asexual. There is no genetic recombination. So when we look at the other type of cell division, which is called meiosis, it would be sexual. And we recombine the genetic material in the egg and the sperm to produce offspring that are different. But this would be asexual. There's no genetic recombination. And the daughter cells, remember we called them daughter cells. These would be in your notes. The daughter cells are identical genetically to the parent cell. Okay. So remember that the chromosomes have to be, before mitosis can occur, they have to become replicated. So we can have pairs of replicated chromosomes. So they could look like this. This would be an example of a replicated chromosome, right? And this is an example of a replicated chromosome. Okay. This would be a pair of replicated chromosomes. That's replicated chromosomes, right? So this happens prior to mitosis occurring. It occurs in S. Remember, we talked about how you go from unreplicated to replicated in S. Okay, so when we look at mitosis, there are 4 phases. And your book goes into more detail about these phases than you need to know. Okay, I didn't actually write that textbook. I don't know why they put my name on it. But it is an open educational resource that everybody contributes to. And then I just kind of put everything together how I wanted it in the book. Okay, so when we look at the 4 phases, you need to know prophase. This is the first phase. Okay, so in this phase, the chromatin condenses into chromosomes. That's a chromatin. Chromatin condenses into chromosomes.
These are the 4 stages of mitosis, right?
Yep. The nucleus has to be disassembled. So what it means is it's taken apart, but the components are still remain, because it's going to have to build the nucleus back at the end of mitosis. So it's broken down, it's disassembled. And it's important that the nucleus goes away, because the genetic material has to be pulled to opposite ends of the cell. Okay. So that's prophase. Metaphase
think middle. Pro means first, meta means middle. So this is where the replicated chromosomes line up in the middle of the cell. Okay. More detail in your book, but this is what you need to know. Anaphase. This is where the cell's cytoskeleton, remember we talked about the centrioles, the cytoskeleton actually pulls the replicated chromosomes apart. So the cytoskeleton pulls the replicated chromosomes apart. That's the third. And then telo. Think telo means at the very end.
So is that stage the one that's held by the centrosomes?
Yes, so the centrosomes would be that part of the cytoskeleton. Telo means at the end. So we saw that word previously in telomere, which was the end of the chromosomes. So in telophase, this is where the nucleus or the nuclei now, because there's two of them, are reassembled around each of the two sets of genetic material. So let's put each set of chromosomes. And at the very end of telophase, we have something that is called cytokinesis. So cytokinesis, kinesis means to break apart. Cyto, anytime you see that word cyte, you know that that means cell. So we talked about osteocytes, chondrocytes, right? Cyte means cell. So cytokinesis is the separation of the cell. So it's the separation of the cytoplasm. So that's cytokinesis. Okay. So we're going to be looking at this in lab today, and we're also going to be looking at how the chromosomes behave. And you have to kind of remember that this is, these are kind of like stages, but there's like in between, because it's kind of like a dance. It's kind of like a set of things that are happening. And so like in your book, they talk about pre-metaphase and post-metaphase, for example. But so it's kind of a continuous thing that is happening. And I'll show you an image of that in a second. Okay, so this is the handout or the diagram, the table in your book that you should look at, right? So notice in this particular instance, they haven't color-coded the chromosomes because this is asexual reproduction, right? So all of my cells, for example, will be have my maternal kind of imprint in them, but I will have still gotten half my genetic material from my mom and half from my father, okay? So you need to kind of be able to identify the different phases. So I could give you this picture right here, and you would see that the chromosomes are being pulled apart, and you would say, oh, that is anaphase. Okay. So these images will appear on the midterm exam. Okay. So, I wanted to show you. So remember that in that picture, they were only showing like maybe 3 or 4 chromosomes, but we actually have what? We have 23 pairs of chromosomes. And so it is much more complicated than it looks like in that particular instance. And let me see if I can find mine. Oh, I don't have it. Let's see. Oh. Okay, so we have a lot more genetic material than is typically shown in the diagrams. So right here, you can see that these would be the chromosomes, which have condensed into the set chromatin, excuse me, that's condensed into chromosomes. And then you can watch how they behave during the process of meiosis. So let's see if I can get this. I don't think there's sound with this particular video. Okay. So they're going to go and they're going to line up in the middle. So that would have been prophase when they weren't lined up in the middle. So they find themselves lining up in the middle. So the chromosomes at this point are replicated, right? And there's anaphase. So they're all getting pulled apart to opposite ends of the cell. Right? And then the nucleus is reassembled, right? And they're showing here just a picture of it. They show the reassembling of the nucleus. Actually, they're showing multiple, actually in this particular instance, they're showing multiple stages of mitosis. So this is what mitosis actually looks like underneath a microscope, an electron microscope, I believe. Okay, stop there. Okay, so now we need to talk about how the body regulates the cell cycle. So when do cells divide and when do they not? Because we don't want cells to divide inappropriately. And when they do divide inappropriately, that is what we refer to as cancer, okay? So we have to have proper regulation of the cell cycle. So we have internal mechanisms which are called intrinsic regulation. So intrinsic means inside, right? So this is how it regulates whether or not the cell is going to divide internally, right? And in your textbook, they talk about the role of cyclin. So cyclin is a protein that is produced by the cell. So it's produced by a cell, and it binds to an enzyme, which is the cyclin, let me get this right, cyclin-dependent kinases. So this binds to cyclin-dependent kinases. Okay. So this is an enzyme. This enzyme is necessary for cell division to occur. So to proceed, we'll say. Okay. So these are abbreviated as CDK. And there's different types of cyclin-dependent kinases. That's a D. And they are numbered like 1 through 4, I think, in your textbook. So if we look at this diagram right here, oops. We look at this diagram. Oh, I need like a third arm. I had this so it was staying put, and then it just kind of decided not to. Okay, so when you look at this, you see that there are different cyclins produced at different points in the cell cycle. So the cyclin B actually binds to the cyclin-dependent kinase 1, and it causes that enzyme to become active, and then the cell cycle can proceed. And so just remember, cyclin is a protein that is necessary for regulating internally the cell cycle. So it should be really easy to remember, cyclin. Okay? The cyclin binds to the enzyme and activates it. So if I was to draw what I think that this might look like from the perspective of molecular biology, it would look something like, now I've lost my pen. Okay. So here's my enzyme, and it has a binding site on it. So this would be like CDK1. Okay, so there's my enzyme. Okay. In order for this to become active, cyclin B has to bind here. So this is cyclin B. It activates it. So it is an activator. And then the enzyme becomes active and can do its work in preparing the cell for cell division. Right? So that is just one way that the cell regulates when it will divide. And for example, if the genetic material is too damaged for whatever reason, so let's say, for example, you go outside and you get a really bad sunburn, the cells in your skin will stop dividing. And they do that because one of the cyclins is not produced in enough abundance to initiate this chemical reaction. And so your cells will stop dividing if the DNA becomes too damaged. And that is a defense mechanism to prevent you from getting skin cancer. So your cells actually die, right? And they peel off when you get a sunburn. And that's just it's a defense mechanism to protect your body from cells that might become cancerous. Okay. So this would be the use of cyclin would be intrinsic. Now there is another intrinsic mechanism that is referred to as the mitotic clock. And you might have heard of this mitotic clock because it's getting a lot of press recently. It has to do with the size of your telomeres. So remember, at the end of the chromosomes, you have telomeres. So this would be my telomere. And there'd be one on each side. Telomere. Okay, that's my telomere. And so as the cell starts to undergo cell division, every time it divides, some of the telomeres are lost. So every time a cell undergoes mitosis, right, some of the telomere is lost. Okay. If you have no more telomeres, so it's kind of like a clock counting down, you're ticking down on the amount of telomere you have. Once all the telomere is gone, the cell can no longer divide. Okay, so it's counted down, right? So generally, our cells can divide long enough for us to live a long life, right? But this seems to be the basis for aging. So one explanation for why we age, right? So this is one explanation for why we age.
If it can't divide anymore, then does it die?
No, it could stay, but like if you think about your skin and it replacing itself, after a while the cells won't be able to replace themselves, or maybe wound healing. So if you think about old people, their wounds do not heal as fast, and it probably has to do with the fact that some of their cells are no longer able to divide. Okay, so this is one explanation for why we age. So one of the really interesting things is that we thought that this was just a standard thing that happened to the cells as they got older and as they continued to divide. But now what we found is that there is an enzyme that can add the telomeres back onto the chromosomes. And this enzyme is called telomerase. So telomerase can add telomere back to the chromosome.
So why don't we all get that instead of a pump?
Yeah, exactly. So we have naturally occurring telomerase in our cells, and we'll talk about where it is used appropriately and inappropriately. But one of the things that they've discovered is that certain life events could actually cause a speeding up of telomere loss. So you probably have maybe all known somebody who had like a really bad thing happen to them, like somebody close to them died. And you see them a year later and they look like they've lived 10 years instead of a year. So it's kind of a rapid aging. They look much older, right? And so what they find is that stress causes your telomeres to shorten inappropriately. So if you're really stressed out, your telomeres will start to shorten. But the interesting thing is that they've discovered that there are certain things that can actually increase the size of your telomeres. And so diet is important, exercise is important, and also like meditation seems to be one of those magical things, yoga, right? That seems to increase the size of your telomeres. So your telomeres can shorten, but they also can lengthen, and this is new news, right? And so along with this new news comes the ability to actually test yourself to see what the size of your telomeres would be, right? So you could go to a website.
So increasing the size of it makes it last longer?
Yes, yep. So the cells are more able to divide and make repairs, for example, okay? So this is just a website called, this is just one of a few out there right now. So you could actually go to the website and pay for a little kit. And generally what it is spit. So they ask you to spit into a test tube and then they take it into their lab and they look to see what the size of your telomeres are, right? And so
filling up one of those test tubes with spit is actually harder than you think.
it is, yes.
I just did that.
I haven't done this one, but I did the 23andMe.
That's what I did.
Right, yeah.
And that was really hard.
So I thought this had a video. Let me see if I have a video with it. Let's see how it works. Get started. Oh, anyway, so this is actually, they use blood in this particular instance. So the really interesting thing is they're now looking at like what is your telomere size before versus after changing your lifestyle. And so in the news, they had this twin study with astronauts. So you might have heard of this. It was big kind of news media. And so they had a hypothesis and they thought that going out in outer space would be stressful, right? So they assumed that the twin that was in outer space for a year would actually have shorter telomeres than the twin that was not going in outer space, right? So they did it before and then they did after. And interestingly, they found that living in outer space actually, for some reason, increased the telomere size. So it was kind of an interesting study, but it didn't last long. So after living, on the planet for a couple of months, the telomeres actually went back to their normal size.
So did they take into account the variable that the other twin or could have just had a really stressful year?
I guess they probably tried to make his life not so stressful. That's a good point. But they also looked at his telomeres before going in outer space and after. And so his telomeres actually lengthened while he was in outer space. Yeah. Which was totally opposite of what they had hypothesized would happen. And they don't understand why that would be the case.
So are there only 2 telomeres on each, or?
On each chromosome, there's 2 telomeres.
On every chromosome.
On every chromosome, there's
There are
2
ends.
telomeres. Yep, the ends of the chromosomes are the telomeres. Okay. They've also found that stressful events in childhood also can cause people to age faster. So if you have a stressful, childhood, you're kind of starting out with telomeres that are, kind of artificially shortened for your age, right? And so that might explain why people that live in maybe poverty or don't have access to healthcare or in stressful environments
might actually die younger than people who have a different type of environment in which they grow up. Okay. So the mitotic clock is a really cool thing that we've just recently started to actually investigate. Okay. So these would be both examples of intrinsic regulation of cell division, but we also have extrinsic. Okay, so this is still under cell regulation, right? Extrinsic means outside, right? Specifically outside the cell. Okay? So it's not like outside in your environment, right? But when we're looking at the chromosomes, they're inside our cell. So extrinsic would be outside the cell. And so an example of this would be, oh, why did that do that? Oh my God. Okay, so extrinsic would be, an example of this would be hormones. So hormones can bind to receptors on the outside of the cell and can cause cell division to occur. Okay. A really good example of this is growth hormone, which is produced by your pituitary gland. It is actually not a steroid, but it causes your bones to lengthen and get longer. So if you have lots of growth hormone, you can get much taller than if you have a little bit of growth hormone. Okay? So growth hormone can initiate cell division in the bones. Growth hormone, that was my own fault. Okay. So example would be growth hormone in the bones.
That way everyone's gonna height the different amount of growth hormone in them.
Yes, and there's actually an interest in parents that have short boys, for example. They want to give them extra growth hormone to make them grow taller because tall tends to be better.
So does that mean, you know, they always say like, if your parents are tall, you're gonna be tall, does that depend?
That's genetic too. Yeah, so the amount of growth hormone. Some people, you know, those people that are gigantic, you know, you could have a tumor in your pituitary gland that causes you to produce way too much, and then you get these people that are 8 feet tall. So way too much growth hormone. Okay, another example of this is estrogen. So estrogen is known for causing cells to undergo cell division. So like the cells that are in the uterus during a particular part of a female's reproductive cycle, those cells in the uterus will divide, okay? So they'll undergo cell division. And estrogen in our environment, we have some chemicals that mimic estrogen, and one of those was that BPA. So we talked about BPA when we were talking about persistent organic pollutants, and BPA is found in plastic, and it leaches out of plastic, and it is actually an estrogen mimic. So another example of this is BPA in plastic, which is an estrogen mimic. So it binds to the same receptors as estrogen. So it's kind of been linked to bad things, specifically in babies, but also could be linked to reproductive cancers in females. Okay, so that is extrinsic. Okay, so we talked about mitosis, and it's really important to realize that mitosis is cell division, but sometimes we actually need cells to die. And so this is what is referred to as apoptosis. So it's kind of the opposite of mitosis. So instead of dividing, cells undergo programmed cell death. So this is programmed cell death, apoptosis. Sometimes people call it cell suicide. right? So it's not your white blood cells attacking cells and killing them. It's actually, there's a switch inside a cell that tells it to release enzymes, and it actually digests itself from the inside out, right? So enzymes are released that digest it from the inside out.
So is that what happens with like cancer cells when your body recognizes that it's
No,
a healthy thing?
no. So cancer cells do not commit suicide. They have to be attacked and killed. So this is not cancer cells. In fact, cancer cells never undergo apoptosis, which is why it's bad, right? Because you want your cells to undergo apoptosis. So, for example, this happens in embryonic development. So certain structures develop early on that subsequently have to kind of be reformed. And one example would be the webbing between your fingers. So this is just one example, right? So the cells that make up the webbing actually undergo apoptosis, and then this creates separate fingers. Sometimes people are born with their fingers still stuck together and there's still web between them, and then they just cut it, right? But that would be an example of apoptosis during embryonic development. We have apoptosis occurring when cells become damaged. So it's protective. So if the genetic material is damaged for whatever reason, it could be from nuclear radiation, right? So you hear people getting exposed to like radioactivity, and then their hair falls out, right? Those are cells that are undergoing apoptosis, okay? So if the genetic material is damaged, the cell will kill itself to protect the organism, the whole organism. So there's actually lots of, if you think about our bodies as being these multicellular, the cells really have to cooperate, right? And they have to be very altruistic, right? So, you know, like our muscle cells can't overgrow, right? That would be bad. They have to stay where they're supposed to be, right? So all the cells have to cooperate with one another. And then if a cell becomes too damaged, it will automatically kill itself in order to protect the body. So that's an important mechanism that we really don't understand very well, that we're just realizing apoptosis is really important. One place that we realize it's important is the immune system, because we could produce immune cells that would attack normal healthy tissue and cause autoimmune diseases. So maybe autoimmune diseases are where something wrong with apoptosis. So the cells that normally would die are able to proliferate and then attack our body and not protect it. Okay, so this comes along with our idea of cancer. So cancer is the unregulated cell division. Okay. So cells divide prolifically, and they also divide uncontrollably. And so if we look at some characteristics of cancer cells, when we look at them, they have abnormal nuclei. I think I showed you a picture of cervical cells that were precancerous. So when you get a pap smear, they take a sample of your cells and they're looking for abnormal nuclei, because that tells them that the cells are becoming abnormal and could possibly become cancerous. Okay. We also get genetic mutations. So we also we'll talk about what these mutations are, what exactly they are. Okay, so they have genetic mutations. Okay. They also have no mitotic clock. So this is really amazing because they essentially become immortal. They do not die. So they can divide and divide and divide and divide over and over and over again, and they do not die. Okay. So I'll talk about an example of that in the lab in a second. Okay. So they do not undergo apoptosis, so they have no apoptosis. That's O, A-P-O-P, apoptosis. Okay. They also have no contact inhibition. So normal cells, if they get in a crowded condition, they will stop dividing, right? Cancer cells, when they get crowded, they just continue to divide, and they get what are called tumors. Now we have tumors that are benign, right? But what is it if the tumor is full of cancer cells? What's it called? It's called malignant, right? So this would be benign is not cancerous versus malignant, which is. Right. So when they take a biopsy, they're looking at the cells and they're looking at, do they have abnormal nuclei? Do they look weird? You know, if they look weird, then they're cancerous. If they look more like normal cells, then it probably is a benign tumor. They also become unspecialized. So, for example, you could have a specialized skin cell becoming unspecialized, and that means that it could migrate and end up in your brain, right? So you could have skin cells that become cancerous, they become unspecialized, and then they can live in the brain. So this means that they can live anywhere in the body. And then the last thing that I want to mention is they can get into the circulatory system. And once they get into the circulatory system, it's called metastasized or metastasis. Okay, they get into the circulation. And so to get a diagnosis that your cancer has metastasized is really bad, because that means that they can't just go in and cut the tumor out, or they can't just radiate a little bit of your body to kill the cells, but they have to do body-wide. So generally they have to put chemicals in your blood, chemotherapy, and that goes everywhere because these can end up in any part of your body. Okay. Also, when it gets into your lymphatic system, that's also bad because the lymphatic system goes throughout your body too. So the reason why we know so much about cancer cells is because of a group of cells that are called HeLa cells. And you might have heard of HeLa cells recently because there's a new movie out about HeLa cells and about the person that donated, unbeknownst to her, her cancer cells, her cervical cancer cells, to the medical establishment. So HeLa stands for Henrietta Lacks. And I believe it was like in 1953. She was young. She was maybe in her 30s, early 30s. But in 1953, she was diagnosed with cervical cancer, and cervical cancer cells were removed. Cervical. That's cancer, cells removed and used in research. So before this time, it was very difficult to grow cells in the lab outside the body. And so they were so happy that they had these HeLa cells to study because they would just grow and grow and grow. And then, you know, one scientist, actually they were selling them. So one scientist would grow a bunch and then ship them off. And they ended up being shipped worldwide to all of these different labs.
And the interesting thing about them is that they were so prolific that they took over all of the other cell lines in the lab and kind of caused this mass hysteria amongst scientists. So like if I was somebody in England and I was studying what I thought to be normal pancreatic cells, I find out that the HeLa cells have invaded my cells, my pancreas cells that I'm studying, right? And all this time I've been studying HeLa cells and not normal pancreas cells, right? So it's like huge problem. So people actually had to go back and rewrite research papers and say, oops, we made a mistake. We weren't studying normal cells, we were actually studying HeLa cells.
So they wrote this really wonderful book called The Immortal Life of Henrietta Lacks. And so this is kind of like science investigation. And the problem is back in that day, you didn't have to have permission to use cells that were removed from a person. And so Henrietta was a patient at the hospital, and she was also African-American and poor. So she wasn't actually paying for her procedures, and she eventually died. And then eventually came out that we were using her cells extensively in research without the knowledge of her family, right? And so this is actually a really great book because it talks about the way that the medical establishment kind of did its work early on and how we've kind of really changed that up to be advocates for patients, okay? So I'm gonna show you a little video that they actually created a movie recently that you can watch if you don't want to read the book and you have some free time on your hands, right? You can watch the video. And so this is just the trailer for this video.
For years, it seemed like a dream. About our mother, could this be true? But you don't understand this? We didn't know nothing about nothing.
Scientists have been trying to get cells to grow outside of the human body, but they would always die. Until Henrietta's cells came along.
I want to write a book about your mother.
Go HeLa, go HeLa. That's my mother. Oh, I don't regret this.
Everyone's saying Henrietta Lacks donated themselves. She didn't donate nothing. They took them and didn't ask.
The hospital is guilty and somebody's going to pay.
God wanna have a disease cure, he provide one for himself.
I don't know, Rebecca.
I'm not doing anything behind your back. Henrietta helped develop the AIDS cocktail, chemotherapy treatments.
It's hard not to get caught up in hope when you've been powerless for so long. I know I'm a part of you, and you're a part of me.
Okay. So, from a sociological perspective, it is an interesting story of kind of race and poverty and the medical community. Any questions about that? Oops, and another.
Is it out in theaters?
I think it's on Amazon. It's an HBO. Yeah, so you can buy it. Hmm?
It's an HBO special.
It's an HBO special, yeah. Okay, so how many of you had actually heard of HeLa cells before today? Okay.
Actually, in the mental health field, that story is kind of common. Because for the same reason, is because in the mental, when they had a psychiatric facility, they would
Yes, so they would use patients as experiments. Yeah, so this book actually talks about that too.
Right.
Yeah, it talks about all the experiments that they used to be able to do.
But similar stories.
Right, yep. Okay. So hopefully we've come a long ways, we don't know. Okay, so I want to talk about some of the genes that are associated with cancer. And so it's important to realize that we have genes that help to regulate cell division. And sometimes those genes can be mutated so that they become defective. And so when we look at a type of gene that helps to regulate cell reproduction, it is referred to as an oncogene. So think onco, think oncology. That is the study of cancer, right? So oncogenes, okay, code for proteins that promote cell division. We have in our bodies what are called proto-oncogenes. So these are the normal form. So this is the normal version of an oncogene. Now, I can be predisposed to get cancer if I inherit one bad copy of a proto-oncogene, right? So if I inherit one bad copy of a gene, so this is just theoretically, right? Inherit a bad proto-oncogene from my father, could be from my mother, but from my father in this particular instance, right? I am at a greater risk.
So what you're saying
For
is that
cancer.
we all have those genes? It's
Yes.
just whether or not we have a bad gene.
Is whether or not if you have the normal version or the abnormal version. So like I have two proto-oncogenes, right? I have many more than that, but I would have like two proto-oncogenes, and sometimes I would inherit a mutant or a bad form of that. And so that is going to put me at a greater risk for cancer. But you have to realize that the environment also affects your risk of cancer. So that if I am a smoker, for example, then and I inherit a bad proto-oncogene, then that actually even makes the risk greater. Right? So when we talk about genetic testing and whether or not you are predisposed to cancer, this is what they're looking for. They're looking for, do you have normal proto-oncogenes? Do you have two normal copies? Do you have maybe one defective copy? So for example, when you talk about breast cancer, so some people are predisposed to getting breast cancer. So they would be people that have inherited one bad version of a proto-oncogene, and they are at a much greater risk, right? So sometimes they just go in and they would have their breast removed before actually they get the disease. Their breast tissue can be normal, but the risk is so high, right? And so that's when sometimes females, in case of reproductive cancers, choose to have their reproductive organs removed. Okay.
So how would it be a bad proto-oncogene?
Okay, so if it is a bad proto-oncogene, it actually becomes an oncogene. So they named these genes based upon the fact that they promote cell division. The normal version of it is called a proto-oncogene. But we have mutations in the genes, and we'll talk about how those mutations arise. But we have different forms of a gene, and one of the genes could be broken so that it will promote cell division no matter what. And so it will cause the cells in the breast tissue to just divide uncontrollably, right? And then spread throughout the body.
So proto-oncogene starts out good and normal, but it can be mutated
Yes.
and turn into a bad.
Yeah, so it can mutate and become an oncogene, okay? So think what this means. Proto means pre-oncogene, right? This is the good version. This is before it becomes an oncogene.
So oncogenes are actually the bad ones.
Yes. So we're going to be looking at, we're going to be watching a video in lab, the first thing, that talks about a specific type of oncogene and how it could be treated, that particular type of cancer could be treated. Okay, We also have what are called tumor suppressor genes. Okay. These are good. They control for proteins that inhibit cell division. So not promote, but inhibit cell division. And you can have mutated tumor suppressor genes that could then make you at a higher risk for developing certain types of cancer. Like colon cancer is actually caused by a defective tumor suppressor gene. And then smoking and bad diet, other like maybe drinking excessively can actually cause the second tumor suppressor gene to mutate, and then you have no control over the cell division, and the cells will just start to divide inappropriately. Okay. Okay, so I need to talk about meiosis. So I would normally probably stop here because this is a kind of a bigger topic than I have time for, but you need to know a little bit about it for the lab today. Okay. So meiosis is sexual reproduction. So it's sexual, right? And it is the production of gametes. So where does it occur? Where's the only place in the body that that meiosis occurs? Testes and ovaries. Right? Only in the testes and ovaries. So in this particular instance, we start out with one diploid cell. And I mentioned that this is a germline cell, right? So this diploid cell is special because it can potentially pass its gene on to the next generation. So it's a germline cell. So this is germline. Okay. From this one diploid cell, we're going to get 4 potentially, not always, but we can potentially get 4 haploid cells. Four gametes. Now in females, we'll talk about that doesn't happen because we produce one giant egg cell and that's it. But we'll talk about that after the midterm. Okay, so when we look at meiosis, we need to have 2 sequential cell divisions. So one cell division right after the other. And so they are called meiosis 1, and I'll leave a little room there, and meiosis 2. So meiosis 1 and meiosis 2. So meiosis 1 divides one cell division, and then meiosis 2 would be the subsequent cell division, right? So actually, let me draw that little picture here. So we start out with one diploid cell, right? It divides into 2. And then this subsequently divides into 2. And this one divides into 2. Okay. So this would be meiosis 1, and this is meiosis 2. Now in meiosis 1 and meiosis 2, we have prophase, metaphase, anaphase, and telophase, but it's always with prophase 1, right? Metaphase 1, anaphase 1, and telophase 1, right? So anytime you see, oh, metaphase 1, you know that it has to be meiosis, right? Because if it's just metaphase, then it's mitosis. If it's metaphase 1 or metaphase 2, so you can put down here prophase 2, metaphase 2, anaphase 2, telophase 2, right? If you know that it has a 1 or a 2, then you know we have to be talking about meiosis and not mitosis. Now, the big difference between mitosis and meiosis is right here in metaphase 1. So I'm putting a little asterisk there. This is really important because instead of the individual replicated chromosomes lining up in the middle of the cell, we have the chromosomal pairs line up. So I'll put the replicated chromosome pairs line up in the center of the cell. Okay. So that means that the pairs line up. So that means that after this division, meiosis 1, the chromosomes are still replicated, right? Because we haven't pulled the replicated chromosomes apart. So the chromosomes are still replicated here. And then in meiosis 2, we go from replicated to not replicated, right? So if we look at a diagram as to how the chromosomes would look, I'm going to skip this one right here. Okay. Now this shows colors, right? So those are the chromosomes that are inherited from the father, and those are the chromosomes that are inherited from the mother. So in my cell, in my ovary, it's going to undergo meiosis, right? Notice how the replicated pairs line up, and then the second cell has the replicated chromosomes, and then notice that at the very end of meiosis, we get unreplicated, right, chromosomes, and there are no pairs, right? This chromosome is different size compared to this chromosome. So at the end, remember, haploid cells have no pairs of chromosomes. Okay. Okay, so I will stop there for today. And I will see you in a few minutes in lab at 2. And here's your recorder